of all, I'd like to thank Dr. Youssef for the interesting talk. It was really important about stewardship and antibiotic issue in our Middle East country. Actually, I speak in another stream or different stream of pharmaceutical sciences, which is about pharmaceutical analysis and how we minimize the environmental impact of our pharmaceutical, either synthesis or analysis on the environment through application of green analysis on a ternary mixture used for hypertension. As Ms. Co-Chair say, I'm Heba Moustafa from Cairo University, and I'm very pleasant being with you here. My presentation contents will be green analysis, green analytical chemistry principle, the GAC principle. It will also include greening approaches or enabling strategies to implement green analysis. And we'll speak about the general guidelines for green procedures, and we'll talk about the green matrix, or how the tools that we are using to assess the greenness of our method, and we'll make more emphasis on analytical ecoscale. And we'll see an application on chromatographic analysis on a pharmaceutical ternary mixture. Please agree with me first that now, due to both scientific and public concern about the environment and health, most of the researchers try to focus more how to make their methodology more green or more eco-friendly. And this will be progressively through application of green analytical chemistry principles. Let's see what are these 12 principles of green analytical chemistry. They include in situ measurements, they include minimization of waste and waste production and waste decontamination as well, minimize sample size, sample volume, minimize the use of hazardous reagents and use more renewable resources ones, minimize the overall energy of the process, and increase the protection for the operator, which is the analyst or whoever is working with the method. So we have some greening approaches, or to apply these green analytical principles, what is the approaches or the enabling strategies we can do? We can use saving solvent strategies like recycling our solvent or dowonizing all the method itself by using minimum volumes. We can apply direct method with no derivatization or no specific harmful reagents. We can use non-destructive analytical methods. We can use high throughput analysis, use combination technique like LC-MS, LC, NMR, HPLC, NMR, whatever. And also miniaturization of the analytical process as a whole to the nanoscale. We can elimination or simplification of the sample pre-treatment steps. Always use online at-line pre-treatment or sampling or monitoring. Chemometric tools if it is possible to have maximum extraction of information from the given data. Also use green or benign solvents and derivatizing agents. To summarize up and to make it easier, we have 4 general guidelines. We have the reagents, the method, energy, and waste. For the reagent, we need to eliminate or reduce as much as possible and replace with more renewable or more benign or more green solvent. For method, it should be miniaturized and non-destructive. Also, it has to be direct to avoid derivatization steps. For energy, we need to less the energy consumption. And for waste, either to eliminate or reduce for the worst case, and have a policy or a tactic for recycling or waste management. Then we try to apply these strategies to follow the green analytical chemistry principle. After when we did so, is our method is green or not green? Then we need to assess our greenness of our method. I'm more speaking about analytical methods, but these assessment tools can be used for synthesis, whatever stage in the drug synthesis or drug industry field as a whole. Green metrics, which mean that we have to assess the environmental impact of any used approach and evaluate it by its adherence to the green analytical chemistry principle or not. From this perspective, we have several analytical tools to measure the greenness, either qualitative or quantitative. And these analytical chemistry metrics or the greenness tool, they should provide us with measurement that covers the 12 principles we talked about, the energy, the waste, the contamination, the reagents. We'll have some of these tools, let's just tell you some of them. Life cycle assessment, LCA, which is a cradle-to-grave assessment. It assesses the whole process from A to Z, from the raw material extraction, production, transportation, even and the ending fate of everything we use in the drug industry. It could be applied to compare the different chemical methods. For example, Glaxo and AstraZeneca have integrated the LCA to its solvent selection and assessment guides, Also, Pfizer performed the LCA assessment for the different processes while producing the lustral, the sertraline, the famous drug. We have also E factor, which is environmental factor, which is a real amount of waste in the process divided by the product, which the ideal factor for a green analysis should be 0, and we don't have these ideals. We have something called atom economy, which highlight the amount of generated waste to yield 1 kilogram of the product. We have the environmental quotient, which is like the environmental factor, but it took into consideration the nature or the type of hazard, not only the quantity. So it multiplied the environmental factor with the environmental quotient of each hazard. We have also reaction mass efficiency, RME, which deals actually more with the stoichiometric ratio of the used substance, not only the mass. We have effective mass yield, which is a percentage of the mass of the product relative to all the non-benign materials used. I'll focus more on the eco-scale, because all of the previous ones mainly used in synthesis, while eco-scale also was previously by Atkin et al. Proposed for assess the greenness of its organic synthesis compound. Later on comes Galuska et al. Suggested an analog of it and called analytical eco-scale that can assess the greenness of analysis, not the synthesis. And it compares the different parameters and steps in the analytical procedures. It is calculated by allocating penalty point. We put penalties for everything in the analysis process that deviate from the green analytical chemistry principle. The perfect green analytical method should have an eco-scale value of 100, which is also the ideal word, we don't have it. We assess mainly for different things or parameters when calculating an eco-scale: amount of hazards, amount of reagents, the hazard itself, energy and waste. And penalty points are assigned for each of these parameters if it departs from the ideal values. So if the analytical eco-scale is 100, it's ideal. If it's more than 75, it's excellent green analysis. If it is greater than 50, it's acceptable. Less than 50, it is inadequate. So how to calculate the eco-scale? For reagents, we allocate a penalty point. And the penalty point, we regard the category of the reagent we use. You know, each reagent we are using in our labs have a safety data sheet, which have pictograms on these bottles. These pictograms include the hazards, if it's toxic, whatever, inflammable, whatever it was. Also, we calculate or we consider the amount of this used reagent. So its type or nature and the amount used. Also, the instruments we are using, penalty points are given for the energy consumption, occupational hazards, and waste, either amount and management. So, for example, this table shows us if the different volumes of energy of reagents used. For example, if you used less than 10 milli in your reactions or equations, you will have a penalty point of 1. From 10 to 100, you will have 2, and so on. Also, the energy, if you consume less than or equal 0.1 kilowatt per your sample, so it's 0, and once increase, the penalty points increased. A reagent penalty point, we follow the Global Harmonized System, GHS, which describe each of the used reagent by 2 things. The pictograms, I told you, these pictures on the bottles, and the single word. The single word could either be warning, so it's less harmful, it take a penalty point 1, or danger, which is more harmful, and it will take a penalty point 2. So for example, if we have a silver nitrate in our experiment, it has 4 pictograms, so it will be 4, and it's a single word describing it as dangerous, so it has 2 penalty points, so silver nitrate will have an 8 penalty point. Then we multiply this 8 with a number relative to the amount of silver nitrate we already used. So for example, this is acetic acid, this has 2 pictograms and danger, so its penalty point is 4. Then you will further multiply this 4 according to the amount used of acetic acid. And also these techniques used in analysis or whatever the method you are using, immunoassay, spectropolarimetry, UPLC, whatever, each one has a penalty point as well. This is a pictogram I told you about. I'm sure you know it. Everything it has a pictograms on it with the reagents. And this is the safety data sheet for silver nitrate. Every reagent has its safety data sheet, so we are concerned more about these spectrograms and this single word, which is danger, so we can calculate the penalty point of the reagent. We applied this concept on a real application about analysis of ternary mixtures used for hypertension. And you know, nowadays, many of hypertensive people use more than single antihypertensive agents. And the fixed dose combination was proved to be faster and easier for the patient. And in the last 10 to 20 years, you have been noticed that in the market, the triple therapy bill which is more common, which is mainly amlodipine, for example, with hydrochlorosizide, with an angiotensin II blocker, telmisartan, valsartan, olmesartan. So this triple therapy combination proved to lower the blood pressure fastly and decrease the obtained or minimized side effects of the drugs if each one is taken alone. Our mixture is telmisartan, hydrochlorosizide, and amlodipine. We work on the chromatographic separation. We adjust the condition and optimize it, putting into consideration the green analytical chemistry principles. We played more, our focal point was the reagents or the solvent. We use ethanol and water. Water is the most green solvent, whatever in the world. So ethanol, it still has a hazardous effect, but it's way less than methanol and acetonitrile, the most common solvents used for chromatographic separation. So we avoid these two hazardous solvents. And we have a small flow rate, so 0.6 milli per minute, and all the separation was done at room temperature. So in our method, we reduce the use of hazard reagents, we reduce the solvent, it is a direct method, no derivatization, we reduce the generated waste, and we have a good separation without affecting the analytical performance of the method. To make sure that our greenness concept does not affect the method, we make the linearity studies with the range, accuracy, and LOD and precision. It was very good results compared to the official methods. Also, we calculate the system suitability for capacity factor, symmetry factor, resolution, selectivity. It was good compared to the reference values. And we compare the eco scale. We calculate an eco scale for all points, comparing it with the two reported methods for this mixture. It have been reported by a UPLC method and another HPLC method. The UPLC method, although it is more encouraging to use a UPLC method from the green point of view, but this UPL method used a sodium perchlorate, used methanol, orthophosphoric acid, and acetonitrile. By the penalty point, its penalty point reaches to 45 points, so its eco scale is 55. The other reported HPLC, it used methanol, orthophosphoric acid, acetonitrile, and trimethylamine, and we calculate the penalty point for the method, so its eco scale of this method is 63, while ours we don't use except ethanol in few amounts and water, which has 0 penalty point, and we have an eco scale of 91. So although we use HPLC method, which is compared to the UPLC method, could be considered it is less environmentally green, but we can use the apparatus or instruments in our lab, so it apply or it present a greener alternate in labs with less expensive and less modernized instruments. So we conclude that we can have a sensitive, accurate, precise, reversed phase HPLC method for green analysis of this ternary mixture, and development of eco-friendly HPLC by avoiding the use of methanol or acetonitrile, the most commonly harmful co-solvents. Analytical eco-scale could be used to assess the greenness of the pharmaceutical analytical method. It also was found that it is greener and safer compared to the UPLC, as I told you, so it can be a good alternate in labs lacking the more advanced instruments. We have a small take-home messages. Apply great analytical chemistry principle, which will be a safeguard for a remarkable increase in analytical method greenness. It is recommended to evaluate your method by more than one tool, and greenness is not it's not measured by just one factor. It's a holistic tactics. It's overall. It's if you have, for example, you have to use certain reagent, no other alternate you can use. Try to minimize the volume of sample. Try to minimize the energy used. Try to minimize other agents so they equate each other or equilibrate each other. So it is a whole overall process. Finally, this is our references, and I'd like you to think green and work green. Thank you.
Thank you, Dr. Heba.